Hi everyone, my name is Ali and I'm a doctoral candidate at the University of Oxford. I'm excited to share with you today my research into the preservation of neural tissues in the archaeological record and their potential as a rich reservoir of biomolecular information. So preserved brains, rather than being unique or exceptionally rare phenomena as is so often claimed, may actually be amongst the most commonly preserved soft tissues in the archaeological record. Reviewing the published literature, I've identified just under 200 reports in 10 languages, which describe more than 1,800 brains excavated worldwide in the last 4 centuries. The central nervous system of a range of fauna and hundreds, thousands, even millions of years old is preserved in wet and dry, hot and cold, aerobic and anaerobic environments on every continent. Yet less than 1% of these ancient brains have been investigated biochemically, and the means by which they persist when other organs perish remains a mystery. So I'm fascinated to illuminate the molecular mechanisms underpinning this as yet unrecognized form of organic matter diagenesis. Adopting a comprehensive approach, we're looking at DNA, proteins, lipids, and metabolites in concert, using a range of extraction protocols and mass spectrometric analyses to identify diverse chemical classes and pinpoint compounds of interest. Given the time constraints today, I'm just going to be focusing on the protein data, but I really want to advocate for an integrated systems biology approach to multi-omics in archaeology. There are several ways we can approach maximizing recovery of ancient proteins. Year on year, the selectivity and sensitivity of mass spectrometry improves, and we've seen parallel developments in things like peptide fractionation, enrichment, and separation. But the protein digestion step remains dominated by the use of a sole enzyme, trypsin. Although there are a lot of compelling reasons to use trypsin, it cleaves at electrically charged arginine and lysine residues, which would be functionally devastating were they to occur in the membrane-spanning domains of many fascinating proteins key to the central nervous system. These brain proteins instead consist of around 50% hydrophobic residues, like alanine and valine, which are cleaved by, for example, chymotrypsin and elastase. Therefore, we sought to explore the utility of a multi-protease approach. More than 400 proteins were recovered with this strategy, and just under a fifth of all protein IDs are known to be either specifically or highly expressed in brain tissues. Protein families associated with a diverse array of neural cellular structures appear to have been preserved in these archaeological brains. The highest numbers of peptide spectral matches are assigned to myelin-associated proteins, but also preserved are those associated with axonal projections, which conduct nerve impulses away from the cell body, and with dendritic projections, which receive impulses from the axons of other neurons. We also have proteins associated with synaptic junctions, which link the two projections as neurotransmitters are released across this gap. However, the most well-recovered protein from preserved brains, in terms of the number of peptide spectral matches and signal intensity, is myelin basic protein. As the major protein of the central nervous system, this is perhaps not surprising. What is intriguing, however, is that regardless of the recovery strategy and across samples, the sequence coverage is fairly low. Despite the multi-enzymatic approach, we've only yet identified the C-terminal half of the protein. While there are a number of different possible explanations for this, clinically, this sequence is known to be an intrinsically disordered region that binds amyloid beta peptides. Amyloid beta peptides possess a high propensity to self-assemble into toxic fibrils that aggregate in indigestible protein plaques, the formation of which can induce other amyloid beta peptides to spontaneously misfold in a chain reaction, and this is known to occur with other proteins too, like alpha-synuclein, Huntington, tau, and major prion protein. Recall that the membrane-spanning domains of many key central nervous system proteins are composed of around 50% hydrophobic amino acids. Hydrophobic interactions play a central role in protein misfolding, and this pilot data suggests that if the cause of relatively recalcitrant myelin proteins have been exposed by early post-mortem cell damage, these hydrophobic residues might become nucleation sites for a cascade of post-mortem fibrillar aggregation. So this begs the question, might brains be preserved in the archaeological record by a molecular mechanism of plaque formation, akin to that which is observed clinically in the dementias? And if so, might these long-dead brains represent the extreme end members of aging trajectories seen in the living? So I'm really intrigued by this idea, and I hope it's intrigued you too. It's been a pleasure to present today. My name is Ali, and thank you so much for listening.